introduce our speaker today, Dr. Rolf Zinkernagel. He received his degree, MD degree from University of Basel in 1970 and his PhD from Australian National University in 1975. He then joined the John Curtin School of Medical Research in Canberra in 1973 as a research fellow. And that's when he began his collaborations with Peter Doherty on the role of the immune system in protecting mice against viral infections. And more specifically, they centered on the role of T cells in protecting against viral infections. And as all of you know, Dr. Doherty and Professor Zinkernagel were the ones who identified MHC proteins as the basis for self-surveillance, allowing the immune system to distinguish self from non-self. And of course, that's one of the founding principles of how we understand the immune system to work. Needless to say, it's one of the most important discoveries in immunology. That Nobel Prize was in 1996, but in addition to the Nobel Prize, Rolf has numerous other awards to his credit, including the Paul Ehrlich Prize, the Albert Lasker Award. He's a member of the U.S. National Academy of Sciences, the American Academy of Arts and Sciences. He's also a member of the Australian Academy of Sciences, the Royal Society, and many, many other things. But the thing that I like most about him is so down to earth and just a great fellow overall. I met him through my mentor, Dr. Andrew McMichael, many, many years ago. And needless to say, he's one of my scientific heroes. So it's my privilege to present Professor Zinkernagel.
Thanks very much for your very kind introductions. So as for most things, about half is true, you know. So I just have to get that thing going, I think.
Can you use the microphone?
Yeah, that's what I try.
This is Dean Proof.
Right. Thank you. Okay, what I'll try to do is to give you a few examples to what I summarized yesterday. And of course, amongst the three major parameters that immunologists like to study, specificity, tolerance, and memory, specificity is the most important. And why do I say that? Because, you know, specificity is defined by the method you use to measure it, like everything in science. So, if you want to show something, you choose a method that gives you the result you look for. And I think this is particularly true for many immunological studies. And most of these limitations have to do with specificity. And I'll try to get right into the matters and show you what I mean with it. So most of academic immunological studies use DNP, dinitrophenyl, or other nitrophenyl groups as a haptenic moiety, whereas what we really want to know is protective antibody activity or cytotoxic or, let's say, therapeutic type of activities. And therefore, the distinction between a serotype definition that basically correlates with protective capacity and the ELISA measurement, I think, is of utmost importance. And I'll give you a example. And that, of course, impinges on what we call cross-protective antibodies, and what that means, and the consequences for vaccines. And I will also talk about T-help within that context. So let's take a T-cell response, of which we know you need peptides presented correctly on a presentor molecule of MHC class 1. Now, with the virus we like to use, which is a non-cytopathic virus, so no direct damage, a nude mouse without, let's say, T cells, and basically survives the infection happily, but cannot clear the virus. So all the immune response does actually is kill all the infected cells that otherwise would not have been destroyed. And what happens in a mouse, if you put that infection into a foot pad on day 0, you get no swelling of the foot pad. So you have a replication of the virus locally, but nothing happens up to about day 6 or 7. And then the foot pad swells like hell, going, let's say, from a normal black 6 foot pad of 1.8 millimeter up to 3.6 or 4.5. So it's really huge swelling. And that swelling reaction here is all CD8 dependent. So if you take out the CD8, the cytotoxic T killers, nothing happens. But usually you have a second shoulder, a bit delayed, and this is CD4 cells. So if you take the CD4s out, this shoulder disappears. So it's a very cheap, easy readout where you actually can kinetically follow the T cell responses. And this, of course, as I've said, is all immunopathology within the foot pad. And if you don't have lymph nodes, where there is no draining of the virus to the draining lymph node, you will not have foot pad swelling because there is no immune response induced. Now, one of the doctoral students in the lab cloned a cytotoxic T cell line specific for that virus, specific for the H2DB presenter molecule presenting a peptide from the glycoprotein of the virus, which is called 33-241. So he made that T cell receptor transgenic mouse. So you now have, we had 2 or 3 strains, founder strains, and these mice, of course, have a much increased frequency of precursor T cells with that specificity. Normally, in a normal mouse, you would have about 1 in 10 to the 6. And if you immunize, you would go up to 1 in 10 to the 5. But that's about it. Maybe temporarily at 6 or 8 days when you see that peak activity, you would be 1 in 10 to the 4. But immediately this frequency would drop down to, again, 1 in 10 to the 5 or so. In these T cell receptor transgenic mice, however, you have about 1 in 2 to 1 in 5 of your CD8 T cells have that transgenic receptor. So, you know, knowing this easy readout, we thought, well, we may as well check this readout in the T cell receptor transgenic mouse. And the prediction would have been that, like in a TB granuloma, where you use the skin test to diagnose your T cell activation in a delayed type hypersensitivity reaction, that now in this mouse, the swelling reaction would be much earlier and, of course, would be phenomenal. So let's do the experiment. We take the mouse, we inject it into the foot pad. In normal mouse, you get this swelling reaction. And now we take the T cell receptor transgenic mouse. And what we found is that, in fact, there's a slightly earlier peak. It already starts around day 3 to day 3.5. But the peak was much lower than in the unmanipulated mouse. Unfortunately, I did these experiments myself because it's, you know, I mean, foot pad swelling measurements, sort of a rather tricky thing. And when you look at regulatory T cells or suppressor T cell types of experiments, people like to use foot pad swelling because you squeeze a bit more, then it's less thick, or you squeeze less, then your foot pad is thicker. So it's, you get what you want. But in this case, now very interesting. Actually, the foot pad swelling was very minor, 20%, which is not very much really. But within a day or two, it dropped off to virtually zero. Fortunately enough, I continued measuring. And fair enough, after day 6, it suddenly came up again. And there were nice 2 peaks. And if you translate that to the original one, this must be CD8 and this must be CD4. And we then confirmed that. But now, question is, why? Why is it that you have this additional peak and then these rather unexpected? Because, prediction was the virus was gone, therefore immunopathology was gone, therefore nothing should be. So, it just smelled like this might be a virus variant that was selected by the overwhelming cytotoxic T cell response in the early phase. So we isolated whatever virus was left in that animal, in the foot pad or in circulation. And in fact, we found a mutation, just one, with a mutation within the GP33241 peptide. So, this was the first time, a T-cell selected virus variant was found. Neutralizing antibody variants under the pressure of neutralizing antibodies have been well recognized, particularly in the influenza, drift and shift story. But this, and then, a year or two later, of course, the same was found in HIV. I give you this as an example of how T cells are specific, because it's a biological example. And it simply shows you that, because we certainly didn't predict that outcome, we just had to do the experiment and find something we hadn't predicted. And it certainly shows the exquisite specificity of T cells, because if you take a gamma interferon type of readout, where you don't look for the disease pattern, you would still find gamma interferon by the original T cell if stimulated with the mutant peptide. So, like ELISA versus neutralization, I think cytotoxicity or immunopathology in vivo versus gamma interferon is not the same. And the readout is very important. Let me take a second example. A classical textbook dogma in Janeway everywhere is the so-called carrier-Hampton effect. And the carrier-Hampton effect is defined as follows. If you prime a mouse, let's say with bovine serum albumin, and then you come back with bovine serum albumin, but hooked onto DNP, you get against the DNP a secondary type of response. If you compare that with priming with a wrong protein carrier, then priming in a way with BSA DNP, which gives you a primary readout. Mitchison, Rajewski, classical papers. Now, think of polio. Polio serotype 1, polio serotype 2, polio serotype 3. We all know if you're immune against polio serotype 1 and you get hit by polio serotype 2, you are not better off. You're as susceptible as if you had never seen polio 1. So, this apparent improvement by carrier priming is okay with an academic antigen, but doesn't seem to apply to an infectious agent. And we know polio 1, 2, and 3 share 99% of all T helper cell determinants. So all that T help is primed by polio 1. And now you come back with polio 2 and you don't get an acceleration or an increased titer against, neutralizing titer against polio 2. So why, why is that? So I'm not saying this experiment was wrong. It's simply done correctly, right, and measured right, but in an irrelevant context. So for that reason, we started, and this is was a postdoc from India, Shiv Sharan. We primed mice, let's say, with vesicular stomatitis virus serotype New Jersey. And we knew, VSV Indiana and New Jersey shared 99 whatever % of all nucleoprotein matrix and so on determinants. And then we challenged this mouse with Indiana, just virus. Forget about the DNP at the moment. Or we had mice that had never been seen VSV, and we simply primed with VSV Indiana. So here we would expect a primary neutralizing antibody response against Indiana, of course. But also this mouse that had been primed with VSV New Jersey against Indiana made a strictly primary response. So there's no, improvement, and I can show you this here. When you look at the titers in terms of neutralization, it's the solid lines, whether you have been primed or not primed, the curves are within a 6-hour accuracy identical. However, if you look at an ELISA readout against, let's say, VSV glycoprotein, New Jersey or Indiana, you would see that a primary is down here and the secondary is up here. So if you measure neutralizing antibody, you get a completely different answer from measuring the response with ELISAs. And of course, the only thing that we are interested in is neutralization. So, now, when you use an established method like a neutralization assay or an ELISA assay, and you come up with a completely new answer, people get worried. They say can't be, and certainly sometimes must not be. So we thought of hooking up the two results by simply DNPylating the challenging virus, just slightly so that it still made plaques, it was replicating, and the whole thing. And now we could also measure anti-DNP responses. So you primed or didn't prime, then came back with the virus that was very lightly DNPylated, and you checked again. Both only had a primary neutralization, but the DNP ELISA response, corresponded to the original result. So I use this to just push the argument that it depends on how we measure, and most of the time we measure something correctly, but it's not interesting. We should measure what we measure. Now, to give you a more general type of explanation, I think, is of course why. Why is that difference? And for that, I think we have to look at the antigenic site recognized by these neutralizing antibodies. As I've said yesterday, these antibodies, the neutralizing antibodies, actually recognize the tip of the trimer of the glycoprotein in a multimeric constellation in the outer membrane, in the virus membrane of VSV. But it's true for most of the other viruses. So the site that such an antibody recognizes is slightly bigger than the half diameter of the trimer, because you can't, all monoclonals compete with each other, so you can't place two independently on the same site. So you have a rough idea of the size of this neutralizing antigenic site, and it comprises something, an area between 8 and 15 amino acids, roughly. Now, when you compare that with a dinitrophenyl or nitrophenyl group, in simply size-wise, this is 6 carbon atoms. So that's the equivalent of roughly 1 amino acid. So the size difference is blatant, of course, and therefore one isn't surprised that such a haptenic unit may bind to a antibody binding site in many ways, particularly if your binding affinity or avidity is very low. And your ELISA permits you to measure 10 to the minus 5 or 10 to the minus 6. And we know the binding quality needed for neutralizing antibodies is 10 to the minus 9 to 10 to the minus 10. So there's a huge difference. And therefore, the explanation would be like the following. The B cell frequency against a neutralizing epitope or antigenic site, as defined here, is roughly 10 to the minus 5 to 10 to the minus 6. The frequency of B cells, and this is not us having done that, it's others, against D and P, is about 10 to the minus 2. It's 1 in 100 to 1 in 50. So it's 3 to 4 orders of magnitudes different. And now, of course, T help comes in, because we like to have IgG responses. So it's clear that if your B cell frequency is so high, you need many, many T cells to deal with all these B cells to kick them off. So your T help is always a limiting factor, never the B cell, because the frequency of the B cells is so high. Whereas for neutralizing specific epitopes, biologically specific epitopes, the B cell frequency is so low that T help is never the limiting factor. And the differences are really, very important, in the factor of 10 to the 4 to 10 to the 6. And also when you look at the binding, at the affinity, it's 10 to the minus 9 versus, let's say, 10 to the minus 5 for the DNP like. But also size and so on, of course, explains that. So, I think we have to revise several of the basic concepts simply because they were correctly measured, but with methods that may not be very meaningful. So let me summarize at this point. Antigens are defined by the size, the organization or order, versus the variability of the determinants. HIV and all these viruses simply vary, and therefore you have a problem. But the size also matters. And if you look at, antibody responses of biological importance, like anti-tetanus or so, this is in the order of 1,000, 2,000 amino acids. There are smaller ones. But you see there is a certain limitation. For example, oxytocin, is only 9 amino acids. There is no autoimmune response or autoimmune disease involving oxytocin. But there is one involving thyroglobulin. So size matters. And of course, DNP and NP and these haptens are simply useless. Now, why have immunologists used haptens and bovine serum albumin, lysozyme, and ovalbumin? Well, in the 1910s or 1900s, immunology was all about infectious diseases. No exceptions. But you, of course, could not isolate and purify the antigens you suspected to be involved. Because, you know, biochemistry was lousy, everything was lousy. But then came the chemists and said, well, we can solve your problem, you know. We can easily get a lot, gram-wise, ovalbumin or bovine serum albumin, and we purify it for you, and then you can stick on something very well defined like DNP or NP, and chemically you're pure, and now you can do your immunology. But of course, biology doesn't work like a chemist's mind, and I think the fallacy of immunochemistry actually have been enormous, because it has really influenced a lot of what we have in the textbooks. And I think, you know, it's, of course, if 99% of immunologists do ovalbumin or bovine serum albumin type of immunology, it's not easy to do infectious disease immunology because you're a minority. So the interpretation is, what I've shown you is the exception. You know, viruses and bacteria are the exception. And you have to prove to the reviewer or the editor that this is not the exception. Well, you know, sometimes life is tough. So, let's try and generalize. And this is a bit oversimplified, but, you know, what I try to do is to tell you a cutoff, let's say, at 10 to the minus 8 in terms of affinity. And say, you know, a VSV Indiana neutralizing antibody may be here, 10 to the minus 9 to 10 to the minus 10. Now, this antibody will have a sizable footprint to bind to the glycoprotein trimer, and therefore, at lousy affinities below 10 to the minus 6 or whatever, will bind to many other things. You know, that's why I drew this in a sort of a triangle. So it depends on what your cutoff is. You know, ovalbumin may be actually binding to these neutralizing antibodies. But at lousy affinity, in an ELISA on plastic, you may actually find a binding signal. So, I mean, there are many people who hate this slide because it says that the name specificity actually is a good name. It's an operational word we use, but we have to define what we understand using the word specificity. And it's binding. It has a binding quality. But it also means we have to understand, you know, what is important. And my argument would be, and you all know that, for example, bovine serum albumin is a transporter protein in serum. Many things stick to serum albumin. Ovalbumin the same. So, you know, that binding is measurable, has been measured, and is certainly below 10 to the minus 10. So binding is not something specific and special to antibodies. It's a very general phenomenon. So in serum or in blood or in plasma, you know, immunology only starts once you have overcome all these, let's say, general, non-specific, general types of bindings. And it's just the frosting, the frosting on the cake that is important. Okay, so that's... Now, let's apply that. And as I've said yesterday, if you do viral-specific types of responses, then it all depends on whether you measure via ELISA or neutralizing antibodies. If you take just neutralization as your readout, then the kinetics are very interesting. You know, you immunize here, you get fantastic titers within a few days, and then these titers drop off very rapidly within 30 to 50 days. Actually, you go below the protective level. If you take ELISAs, mouse makes still ELISAs, up to your ears for 300 days. The distinction, of course, is all the quality of what you measure. In one case, 10 to the minus 9. In the other case, 10 to the minus 5. And of course, while this is taken very often as an equivalent for protection, it isn't. The only equivalent is the titer of neutralization in the serum at the time of confrontation with the infection. Now, people working with DNP and NP particularly have shown that all antibody responses undergo what is called somatic hypermutation, or maturation of the affinity over time. This has been particularly well documented for NP because, most of these people like to play around with that nitrophenol. Now, if you look at the kinetics, it takes about 20 to 40 days for such an NP antibody to affinity mature. Of course, you start with 10 to the minus 5 or 6, which is lousy, and you improve it to 10 to the minus 7, which is not bad, but it's still lousy. Now, with neutralization, of course, this is not sufficient. You have to be at 10 to the minus 9 to minus 10 in the mouse. In humans, it's even 10 times higher. And this has to be done in 4 days, because otherwise you're dead. So, is there affinity maturation in the neutralizing antibody response against a virus? We use again the example of a rhabdovirus, VSV, New Jersey and Indiana, and a doctoral student, Hans-Peter Ost in the lab, did fusions at the time, on day 6 after primary infection, or 12 days, or 150 days, that is, after having boosted that response 8 times. And then he made fusions with hybridomas and simply had a spectrum of monoclonals that then were determined as to, affinity and neutralizing capacity. And what you see is that even on day 6, this is all IgG. On day 6, you have a spread between, lower than 10 to the minus 10 to lower than 10, around 10 to the minus 9. And this average spread doesn't improve over any of these immunization schedules. So I cannot, and I emphasize that, I do not exclude that some somatic hypervariation occurs during that time. That's not the issue. But the quality of the neutralizing antibody binding has to be there on day 4. And this is basically reflected in these extremely high values there at very early times. So there is no overall improvement of the response over time. And you also see in terms of neutralizing capacity, there's on average no evidence of making the response more powerful. Another thing that sort of just fell into our lap was, which I thought was rather surprising, and this is Ulrich Kalinke in the lab who did it. At that time, that, sequencing became more easy. That was in the late 90s. And we could actually sequence all these monoclonals and find out, which gave us the highest affinity to start out with, and was there a change in repertoire. And this, in fact, was rather interesting, that most of the initial day 6 usage of heavy chain was rather restricted. And this major group sort of, got diluted out by other families that took over. So it looks as if the initial response is at least oligoclonal. if not almost monoclonal. But it's a very narrow, and this, I think, in a way, looking back, isn't so surprising. You know, how many specificities are there on the antibody repertoire? When you look at the textbooks, you can choose between 10 to the 7 to 10 to the 11. But this is, of course, the thinking of dinitrophenol small antigens. But if you consider pathogens, you know, the spectrum is not that huge. I mean, human pathogens, if maybe on a sunny day, you know, 10 to the 4 is probably already pushing things quite far. So the only thing you need to be able in an operational way to recognize is about 10 to the 4 determinants that are relevant for survival. Maybe 10 to the 5, I mean, let's be generous. But it's not 10 to the 7, it's not 10 to the 10. So with 10 to the 4, you know, and you have a 1 in 10 to the 5 precursor frequency, actually you need 10 to the 6 to 10 to the 7th B cells in a mouse that actually would cover, you know, that spectrum of pathogens. And that at least fits reasonably well with the numbers we found. Now, we used a trick to actually follow where this all happens in the lymph node or the spleen. And, you know, you can easily imagine if a viral particle only exposes on the surface the key neutralizing determinants, nothing else on the intact particle, you can easily think of using the intact viral particle as a probe to look for B cells that recognize that unit, because nothing else is exposed. So that's exactly what we did. So we used purified virus that was harvested very early after initiation of an infection in tissue culture, where you know you have few defective particles, and we used the intact particles to probe for B cells and for plasma cells, because it works like a charm, you know. And then we detected the bound virus, let's say with a neutralizing rabbit antibody that then could be, you know, second stained with some peroxidase or something else. And when you look at that, then you see in a lymph node, this was day 12 after a VSV infection, you see with PNA, you see the germinal centers, you see the FDCs stained with an antibody, and within this, you see VSV-specific B cells stained with this virus probe. And what one sees, actually two types. One sees these dark red solid cells that stain almost entirely. And then one sees cells that show like a ring staining. And the solid cells are plasmacytes, because they stain, of course, in the whole section of the protein in the cytoplasm, and the others are specific B cells. And you then can use that trick to go into kinetics and so on. And we got quite a fairly precise idea, you know, what the kinetics are and when what happens. Now, I stop here on these neutralizing antibody responses, but in the meantime, a postdoc had come from King's College in London, and he was a gastroenterologist and was interested in so-called oral tolerance, which I happen not to believe in, because I think there's no experimental evidence except for mice being gorged with 10 grams of ovalbumin or bovine serum albumin where you can show something to happen. Because I think in practical immunology, there's no evidence. So he wanted to use gastrointestinal bacteria to insert a certain antigen and then follow the immune response against that antigen systemically to look for tolerance. And I had an old interest in IgA for historic reasons and said, well, first let's study what the local IgA response is. And what turned out, to happen after several studies was that, in fact, there is an IgA response in the mucosa, as we all know, but that IgA response actually is independent of the serum IgA response. And we found that because, being in the Department of Pathology, any mouse that crossed our lab, transgenic, knockout, knock-in, whatever, we always worked up completely histologically. Looked, for CD4, CD8, B220, the whole thing, including all the classes of antibodies, IgG, M, D, A, E. And doing this, I had found previously that, funnily enough, these IgM knockout mice, the MUMT knockout mice, had no B cells in the spleen and the lymph node. There was no IgD, there was no IgM, there was no IgG, no IgE, but there were IgA positive plasmacytes in the gut mucosa. That was funny because, after all, it's a knockout. But it was exclusive, the local IgA in the mucosa. And so Andrew McPherson then did the functional studies and found that, in fact, black 6s, let's say, make this local IgA, but the MUMT mice also make this local IgA. So it looks as if this IgA in the mucosa is something rather distinct from the classical immunoglobulins, and smells at least like a rather primordial type of local antibody-like response. And interestingly enough, I don't have the data here, it is independent of Peyer's patches. So the organized secondary lymphatic tissue of the gut mucosa is not necessary for that local IgA production. It's simply dispersed through the mucosa. And I just mentioned this, that, there's plenty to do. If you do experiments, you have a chance of finding things you wouldn't expect. And this certainly was another example in our own hands. So let's try and summarize at this point. What did I do? No. So, if we take IgG, everybody accepts IgG is completely T-help dependent, and you need cognate interaction. Now, if you look at IgM, IgM is completely T-independent. Of course not against academic antigens, because they do not present in the right multimeric repetitive type of structure. IgA, the local mucosal IgA, seems to be completely T-independent. And after all, there's that interesting relationship between IgA and IgM. But then there's serum IgA. What is the role of serum IgA versus the local mucosal IgA? But there we haven't progressed very far. One speculation, of course, would be to say the serum IgA is part of the secretory principle that is important in milk production and protecting the offspring via the milk antibodies. But this is just a speculation. IgE we looked at IgE responses against, under, let's say, quasi-infectious disease conditions by keeping nude mice in a dirty animal house versus a highly, pathogen-free environment. And what we found is that nude mice kept in a conventional environment, had a hyper IgE syndrome within 6 months. They had IgE up to their ears, but no IgG or very little in any of the other classes. So that hyper IgE is apparently completely T-helper independent. In contrast, the specific anti-parasite, anti-worm IgE response, which we also studied, is completely T-help dependent. So I think there are lots of things we don't quite understand yet, so there's plenty of room for younger researchers to go after these open questions. And the textbooks often give this impression that everything is solved. It's not true. You just have to look at something you get interested in, and you always find something interesting. Now, I try to address, regulation, because it's very much in favor in immunology, because regulation is so easy, isn't it? It's like with the financial markets. They do all these crazy things, and then somebody says they must be regulated. But this regulation never works, Doesn't work. Regulation is something that doesn't work. And I think the same is true for the immune responses. The only thing that regulates an immune response is the antigen. The antigen comes from the outside, gets in, gets amplified, that's an infection. Then you induce an immune response. That has to stop a cytopathic infection. The antigen goes, the immune response decays, and that's the end of it. If the infection stays, like with HIV, HBV, HCV, malaria, all these parasitic diseases, then you start accumulating immune complexes and all the mess, and you end up having immunopathology due to persistence of antigen at various levels. So, what were the regulatory types of approaches immunologists have liked? Idiotypic networks. This was a great thing in the 60s and 70s. There was even half a Nobel Prize given to idiotypic networks. And the idea of having an antibody as an antigen that is controlled by an antibody, is intellectually very attractive. I mean, you can do mathematics, modeling, the whole thing. But the evidence isn't there. So I'm not saying, in certain model situations, and Ron Levy treated some B-cell myelomas with anti-idiotypic antibodies, and the anti-idiotypic antibody was quasi a anti-tumor antibody. And it sometimes, in some cases it worked. You could get rid of these B cells producing that antibody. But as a regulatory and regulating concept, it certainly has disappeared. And interestingly, like most of these things, with the death of the proponents. The same I predict, of course, for Tregs. And the only thing that regulates is the antigen dose. And most of the experiments done to show Tregs are very complicated and complex experimental setups. And that, of course, prevents other people too easily to repeat what you have found. And that's, for some time, quite a safe ground not to be caught and to stay with a unique result. But that is not going to solve the issue. I think we need a standard protocol that everybody can repeat, and then everybody can play with it and do control experiments or falsifying experiments, I think, would be even better. Now, I would like to illustrate what I've said that antigen regulates, if I want to use that term, the immune response. And for that, I have these sketches here. Usually what happens is an infection hits you somewhere in the skin or the mucosa, and from there the infection gets to the draining lymph node. There's local proliferation here, then local proliferation here, and then there's usually hematogenic spread. And of course that's the dangerous phase, because then the virus or bacteria or the toxin can get to the brain, and that's the end of it. And this has to be prevented. So the very early IgM response in the draining first lymph node is of utmost importance. But there are other examples of infections. Two extremes are shown here. One is papillomavirus. Papillomavirus infects the terminal cell layer in the epithelial, skin, for example. Now, it does not replicate in that terminal cell layer, because papillomavirus has control mechanisms that lets it proliferate only once keratin is expressed. So the first replication after infection is only when these germinal cells differentiate outwards into keratinocytes. So that basically eliminates that virus infection from any contact with the immune system. Because out here, there are no Langerhans cells, there's nothing, no dendritic cells, no macrophages, nothing. There's no lesion. So papillomavirus basically infects and stays out of the reach of the immune system. And the other extreme is this. As I've said yesterday in the lecture, that there are certain viruses that by definition are non-cytopathic, that get transmitted from a virus-carrying mother to the offspring either before birth or at birth. Hepatitis B, hepatitis C, HIV, and so on are examples. And of course, at that time, the offspring has no immunocompetence, and therefore there is no immunopathology. So the virus just jumps and replicates, and everybody is happy. Now, that means that the T-cell repertoire in the new recipient deals with these infections as if these infections were self. I mean, functionally they are self, while genetically, of course, not. But they behave in terms of distribution, kinetics, life, all like all serum antigens of the host. It's there, it's always circulating, it's everywhere where it has to be, including the thymus. So, it's antigen localization, distribution, and the time that are important. Now, if that is so, and if that is strictly dependent on secondary lymphatic organs, then of course one could argue that if we stick antigens subcutaneously, the transport of that antigen via dendritic cells or monocytes or macrophages to the draining lymph node must be very inefficient. So can we improve performance of an immune response by simply sticking the antigen right into the lymph node or the spleen? And Thomas Kundig did these experiments. So he compared intradermal, intramuscular, into the spleen or into the lymph node, and used, these types of doses. And you can do that with the antigen, or you could do that with UV inactivated virus, or you can do it with DNA, for that matter. And when you do that, you find that intradermal, even with 200 microgram, in this case it was DNA, you barely get an immune response. But if you stick 0.2 1,000 times less of that same material into a lymph node, you get a very potent immune response. So nothing happens outside of the lymph node, and the efficiency difference is just very impressive. So let's do again the experiment I explained to you yesterday. You take tumor cells, single cell suspensions, 2 million, 10 to the 5, doesn't really matter. You inject those subcutaneously, and then you wait for a tumor. Nothing happens. If you take the same cells, you pack them into a sort of a cell package, then always you get a tumor. And the difference is that in this case, you always find a few of the fibroblasts. This is a methylcholantrin-induced tumor cell line with a very strong introduced transgenic viral antigen as a very strong tumor antigen. You see this seems to correlate with a high degree of immune responses. And as I've said yesterday, if you combine the two experiments into one mouse, then always the induction wins and prevents the solid cube to grow. Now let's do this experiment in an even more extreme situation. And that is in a completely allo-antigen incompatible situation. So there's a complete MHC mismatch between donor and recipient. And this is particularly, of course, interested in diabetes. And you know about the Wisconsin protocol that tried to transplant islets, isolated islets, injection through the portal vein, and then in some cases, yes, you got on the immunosuppression, you got take and repair of the diabetes. Now, in this model situation, you can be much more stringent because I'm lazy, so I didn't want to isolate islets, which is really difficult. And the second thing I didn't want to contaminate the relevant cells with anything else that could be mobile. Remember just the previous experiments. If cells could go to the draining lymph node, you had an immune response and rejection. So I used a cell line that had been made by an Israeli group that was of H2K haplotype and had sort of a selecting marker, tetracycline, that you could use to prevent the tumor from growing, too much. So we transplanted these islet cells in a packed version, like I told you with the tumor, the same idea. Prevent any of the cells to get to the draining lymph node or the spleen, and pack them under the kidney capsule. So now you take a black 6 mouse, completely histo-incompatible, and you get rid of all islet cells by streptozotocin treatment. And if you don't do anything, the blood sugar goes up and stays up. But now we transplant these packed islet cells under the kidney capsule. And within 10 to 20 days, blood sugar goes down. This is completely allogeneic. So now, of course, you would expect, the mouse was able to regenerate from its ductal cells some islets, and this is good enough to correct the diabetes. But this is not true, because you have the advantage of simply being able to take out the kidney, and if now the blood sugar went up again, you knew all the correcting whatever activity was in the kidney. So that's what we did, and blood sugar goes up immediately. So here we have a situation that you transplant completely allogeneic insulin-producing cells, and they are not rejected. Now, of course, the next question is, what can I and have I to do to get rid of that transplant? How can I disturb this apparent balance? Is it regulatory T cells? Or, well, if here down, 2 months after transplantation, you now immunize that mouse with C3H spleen cells or lymph node cells or macrophages or dendritic cells, and you immunize once with really pushing, the immune response, nothing happens. The blood sugar stays down. So you use what is assumed to be the strongest immune stimulation in transplantation immunology that you can get, which is full thickness skin graft, let's say of 2 square centimeters. You transplant, you take this corrected mouse, and you transplant here a skin flap of 2 square centimeters. And of course, this is a very interesting experiment because now you can control, is that rejection of a primary type or is it of secondary type? Because if the mouse has been sensitized with the transplanted islet cells, the skin should be rejected in a secondary type. Well, it isn't. The skin is rejected in a primary fashion. So the mouse has not been immunized against the foreign transplantation antigens. Of course, it's epithelial, it's islet cell, so it's only class 1. But still, the mouse is not sensitized. It's a primary type of rejection. And still, the skin comes off in 10 days, as it should in a primary fashion, but the blood sugar stays down. So the islet cells continue to function. I mean, temporarily, you see a small infiltration, but it's not good enough to reject all the functioning islet cells. So what does that tell us? It tells us that transplantation antigens are not extraordinarily strong antigens. And the immune system doesn't primarily react against transplantation antigens. As long as you keep the transplantation antigen foreign outside of the lymph node and the spleen, nothing happens. So you can trick yourself into the system by simply using that idea of geography. But the second, I think, very surprising finding was that even if you hit now the situation with the best inducer of an immune response, like full thickness skin grafts, you couldn't reject the transplanted cells under the kidney capsule. And that, of course, is relevant to our thinking about tumors. Because you could simply argue, in this model situation, this is an epithelial tumor that is strictly outside of lymph nodes and spleen. And once that is established, even with the best immunization protocols, you can't get rid of it. So, and of course, this guarantee simply guarantees that autoimmunity or immunopathology against our own, for example, hormone-producing cells may be induced, but it's always not efficient to kill more than some percentages, but certainly not 95 or 98%. And therefore, the function is guaranteed. And I will now finish with just simply showing you this experiment where we took our virus, which is non-cytopathic, so that's why you can play with it, and you take a very small dose of infection. There you get virus replication, a good CD8 T cell response, always control of the virus below detectable level, whatever that means. It doesn't mean that the virus is completely gone, but the T cell response is slightly higher, and persists. If, however, the same type of mouse you infect with a very high dose of this non-cytopathic virus, there's something else that happens. You get induction of a T cell response. The virus, of course, inundates the whole organism. T cells disappear within 7 days, and you will never see that T cell response coming up again later. And the virus stays at high levels in the blood and everywhere else. Now, the interesting part is the in-between dose. So you get lots of virus, you get a tremendous T cell response, and after about 30 to 60 days, most of the mice die. And they die of AIDS. I mean, virus-induced immunodeficiency and immunopathology. The liver goes kaput, everything, is sort of like in an internal graft versus host, host versus graft. The immunopathology just chews up all the organs where the virus is. And then 95% of the mice die, but you have a few survivors. And when we then, let's say at 100 days or so, check in the survivors, there are only 2 groups. In one, the virus is controlled and you find cytotoxic T cell responses. And then the other group, you find virus but no cytotoxic T cells any longer. So there is no in-between. But the in-between, ends up in a chronic, inflammatory, degenerative type of immunopathology that basically kills you. In humans, this could be MS, it could be chronic lupus, it could be any sort of chronic lung disease with fibrosis, chronic hepatitis with fibrosis, and eventually development of a primary liver cell carcinoma. So, immunology is very simple, I think. Either the antigen doesn't reach the draining lymphatic organs, then nothing happens. Cornea transplants would be of that type. And the papillomavirus initially. Or the infection, is in the periphery, but also reaches draining lymph nodes, influenza, all the mucosal types of infections. And then you, of course, get rejection, like in transplant skins or hearts, and control of influenza or polio. Or the virus infection is in peripheral organs, but also reaches secondary lymphatic organs, but it's never controlled completely, and then it sort of comes up, every once in a while, like herpes viruses or zoster. And it is, of course, this repeat exposure that keeps the immune response running, and that we usually call memory. Although it is, of course, philosophically, it's not memory, it's re-stimulation, periodic re-stimulation. And in the transplantation situation, you call it chronic graft versus host or host versus graft reaction. And in infectious diseases, TB, hepatitis B, HIV, malaria, all these parasitic diseases would be good examples. And then there's a last, where the infection overruns the periphery, but also overruns all the lymphatic, peripheral, tertiary and secondary lymphatic organs, and that leads to immunopathology. Then you are in this category, or to complete deletion of T cells, and then, of course, you have a proper bone marrow chimera, or you have virus infections that are, without chronic inflammation. So, that's it. Immunology, I think, is very easy. It's governed by antigen, the time kinetics, the dose kinetics, and where the antigen is. Thank you very much.
So I'm wondering about these non-neutralizing antibodies that you get in response to a virus. Do you think they serve any biological function, or are they just to confuse immunology?
Well, you know, in biology, in very general terms, it's very difficult to say absolutely nothing. So I wouldn't go that far. But if you take a replacement, approach, neutralizing monoclonal antibodies do the job, period. Whereas monoclonals against nucleoprotein or matrix or any other stuff doesn't do it. So
You don't think it function in antigen presentation?
no evidence. Because remember, I don't believe in cross-presentation. But the evidence is, we did these experiments, it doesn't work.
Robert Bosch from the University of Kiel has made a rather provocative hypothesis that although we like to study the immune response to pathogens, he feels that the immune system has evolved to actually deal with our commensal microbiota. And I was just wondering if you have any, thoughts or reflections on that hypothesis.
Yes, I think the two things are, what we call biota is nothing else than things that usually are not pathogens. So there's no bloody difference. But we did actually, and this is particularly Andrew McPherson, we used mice that were completely sterile, and then we repopulated them, let's say, with 5 defined bacterial strains. And the interesting thing was, we used, one of them was marked with something we could easily follow. Now what happened is, if you take a completely sterile mouse, and you come in with such a bacterium, you find that the first thing that happens, the bacterium is everywhere, not just in the gut where you put it, but all over, systemically distributed. And within about 7 to 10 days, the bacterium in the general circulation and in other organs disappears, is still in the gut. And this goes with the local IgA, specific IgA development. So it looks as if the so-called gut biome is controlled by the local IgA response. So I think, in a way, it's just part of that. And whether you call it a pathogen or not, as you well know, is sometimes dependent on whether it's a sunny day or not.
Thank you, Rolf.